want me to introduce over here? Great. Here?
That's good.
Is that good? This is my good side. I'm glad the residents are here, and I think we'll have more coming, but it's my pleasure today to introduce Dr. Hajianpour, who's our new faculty in genetics. Dr. Hajianpour graduated from the University of Tehran from his medical school. He went and got a PhD in human genetics at the University of Newcastle in the United Kingdom. He then went to Miami and did both a fellowship, residency in pediatrics, and a fellowship in clinical genetics. So he's had a lot of training. He also did a cytogenetics fellowship in Pasadena, California. Dr. Hajianpour worked in California for a number of years with a Genentech and I'm sorry, not Genentech. What's it called? I'm blanking. Well, the cytogenetics. Which one?
Genetics Institute.
The Genetics Institute. Genentech is a company that has products, but he consulted with them and oversaw about 45 genetic counselors working in perinatal genetics, cancer genetics, and pediatrics. So he has a wide range of clinical experience. Then he went to the University of Iowa, and he's come here from the University of Iowa, where he started a fellowship in cytogenetics. And I just think when I first got here, we had very limited genetic services for the entire region. And given the importance in medicine right now of genetics, I thought that was really, really abysmal. So we did a pretty big canvassing of the country and asking people to consider applying to a position, and Dr. Hajianpour stepped up, and he's agreed to kind of build. He has a big vision to build a genetics program for this entire region. And I just was speaking with the chair of OB. We've talked about adult cancer. So we're really looking at how can we build a genetics program out of pediatrics that would service the needs of multiple departments. I really encourage all the residents to take genetics. I know your schedules are packed, but there'll be a day when it's guiding so much of what we do in both rare diseases and common diseases. I mean, there'll be a day when a person comes into your clinic with asthma and you want to know their polymorphism so you can know what drug to take. So this is really good, and I think you've already seen from some of the presentations in your readings that the diseases that we know clinically have so many variations in genetic origin that we really need to be pretty well versed in our genetics to treat kids nowadays. So I really encourage you all to take his elective in the future, and with that, I just really invite you to come on up, and thank you for coming, Dr. Hajianpour.
All right, I'm going to talk on the genetics in the past, which the state we are now, and then the future. And basically, the past, this is just a double helix DNA that was discovered in 1953, but it seems to be, with the pace that we have so fast in genetics, it seems to be the past. And right now we are doing sequencing, the gene sequencing, panel of gene sequencing, whole genome sequencing, exome sequencing, all goes with sequencing, looking at the genes at the molecular level. And by doing that, still we have to dig a lot, and there are a lot of unknowns still to be discovered. And if we go, there's no disclaimer too. If we go very to the past, 13 billion years ago or 14 billion years ago, when there was nothing, if you can imagine nothing, there was space, darkness, and coldness, and then we had the Big Bang. there should be a noise with this.
Oh, no.
Okay. So, maybe this is. Okay. Let's see if it does. No. And basically, the Big Bang, and just imagine the explosion and everything, and the big noise, and eventually we had the formation of different planets and different stars. And after 9 billion years, about 4.5 billion years ago, we had the formation, the Earth just started to form. And basically, we had, after about, the pointer is there. Okay, about 1.5 billion later, we had some organism, organic structures that started to just develop. And another billion years later, we had some prokaryotes. And a billion years later, we had eukaryotes and multiple cells. And about 800 million years later, about 200 million years ago, we had all these animals around dinosaur age, and then eventually 135 million years later, 65 million years ago, we had all the animals in the land, air and ground on the land, and basically another 60 million years later, we had the great apes and basically the start of having the human race. So the human, basically, we're talking about 4.5 million to 5 million years ago. So from this point here, that we had even the prokaryote, up to here, this guy who is surfing, we are talking about 2 billion years. It is not a kind of fast process. So we had basically all these evolution, natural selection, fitness, and everything, that from here to very first cell to Mr. Darwin here, we are talking about 2 billion years. So all these natural selection that we are talking about, or evolution, it was single base by base that happening, and took millions of years. Now, so the Homo sapiens, that which are us, basically is the binomial nomenclature for the only extant human species, and Homo is the human genus, which also includes the Neanderthal and many other extinct species. And basically, Homo sapiens is the only surviving species of the genus Homo. And again, we are talking about from this to here as a human, about 4.5 million years. So if we look at all the different animals, similarity in DNA, we will see that we have all the same origin. Now, even between you and us, 99.5% of genome is similar between us, and only 0.5% difference makes us different from each other. And even chimpanzee, 96 to 98% similarities, the cats 90%, the cows 80%, and 75% of human genes have equivalent in mouse, and the other way around, 99% of mouse genes have equivalence in humans. And that's why we can make mouse model for different disorders that we have, like diabetes, Prader-Willi, or other genetic syndrome, which is basically done in Jackson Laboratory in Maine. And basically, when fruit fly has 60% similarity in DNA, and the source of this data is from Homo gene. Now, not everybody accepts evolution, right? Like a doctor, a surgeon, a neurosurgeon. That basically, that's quote, what he said, no one has ever demonstrated one species changing to another species, so unless you have found the fossilized remains of the elusive lizard man, you can keep your bogus evolution. As if the lizard is going to change from the toe up gradually to human. So in this situation, you just want to scream like this guy, what is he talking about? Or this guy. But no bad words. It can be nice, right? Oh yeah, okay. Our future presidents. Okay. So, but in a more serious way to look at it, this is Francis Collins that he was basically, right now he is the director of NIH since Obama appointed him. And believes in creation and God, and also believes in evolution and Big Bang. So by being, that's what he says, by being outside of nature, God is also outside of space and time. Hence, at the moment of the creation of the universe, God could also have activated evolution with full knowledge of how it would turn out, perhaps even this presentation. The idea that he could both foresee the future and also give us the spirit and free will to carry out our own desire becomes entirely acceptable. So basically, it doesn't matter what you believe in, basically, the evolution is something real and based on all the similarities that I just explained. So let's talk about creation. A long time ago, so God knows when, God created all these animals and humans and Adam and Eve, and you see they are so perfect. They are white, blonde, and probably blue eyes, and that's why they are so perfect. And however, okay, Miss or Mrs. Eve, if they were married, deceived Adam, and they ate forbidden fruit. And God got angry and threw them out, right? When? I don't know, maybe 5 million years ago? Okay, he was so angry. I don't know if God sent a human like this or like one of these and said, go and evolve. You're on your own. But the other possibility is that because he was so perfect, these guys all just were following him, right? Anyway, okay, Mr. Darwin. He presented the theory of evolution and natural selection in 1859. And basically, this is the second half of 19th century, and we will see that everything started from now on. And he was saying that it is not that we survived because we were stronger or more intelligent, but because any species can adapt better to the environment and adapt to the change. So let's go to 10,000 years ago, then when we had the civilization and discoveries and progress, Plato, Aristotle, and Leonardo da Vinci, who was painter, sculpturist, physicist, anatomist, engineer, everything that you can imagine, and he was genius. And Galileo, who introduced the heaven or the skies to us, and Newton, who introduced the gravity to us after studying under the apple tree, and the apple fell down, probably hit the head. And then Mendeleev with the periodic table and change of elements from one to each other based on the number of electrons and protons, and neutron, and basically Edison with hundreds of thousands of inventions, and probably that led to this guy, Einstein, with theory of relativity. And then we have Stephen Hawking, who is a physicist, a theoretical physicist, and the only one who holds the chair after Newton in Cambridge. And then we have Bill Gates with the computer, and Steve Jobs with Apple computer. And this guy has a genetic disorder, as you may know, and he's the one who introduced the Big Bang and black hole and all those, and still giving new theories. And he has a genetic disorder, ALS, amyotrophic lateral sclerosis, a motor neuron disease, that starts usually at age 30, 40s, but he started that at age 20, late 20s, and first with just falling down, and then could not walk, and then could not move, and lost voice, and but it doesn't affect the eyes, it doesn't affect the heart and internal organs. So thanks to these guys, somebody in Caltech, California Institute of Technology, made a computer for him that he can look at the letters or words and choose them, and this way he can talk and write. And he's still doing that. And this guy, Mr. Mendel, John Mendel, who was later on called Gregor Mendel, in 1822, he was born in Austria at the time, now it is Czech Republic. And at age 21, he attended the Abbey just because he didn't have money to attend the school, and they were providing free education and all, mainly physics. Mendel became a priest in 1847, and then 1854 became a teacher over there for physics. In 1856, at age 54, he started his studies. And basically, he was studying 7 characters in the plant, the pea plant. You go to the shape of the plant, the size, the color of the flowers, the seeds, again the size and different characteristics. And based on that, he came up eventually with in 1866, at age 44, he actually published data that basically was saying that the inheritance is through something that now we call genes, and the trait is coming from the parents, and the parents may not have the trait, but the child may have it. And basically, this is the Mendelian kind of inheritance. However, his work was not recognized for 34 years until 1900. And 3 scientists from different areas, they didn't have independently basically doing the studies, and one probably is from England, the Greece from Paris, France, and German. And for some reason, they didn't have internet at that time, and they couldn't communicate well. So, but when they wanted to publish that, they found out that it is already published. And that's when the new science of genetics was born. And Mendel died in 84, 16 years before even the discovery of the Mendel laws. And basically, now we know that most of the plants and animals follow the pattern of inheritance based on Mendel's law. In his honors, we call these inheritance, the mode of inheritance, as Mendelian inheritance. Autosomal dominant, autosomal recessive, X. Now, and then we had hereditary, human genetics, medical genetics, clinical genetics, and then like all the branches of medicine, sub-branches of these specialties, and it's going on and on. Now, by that time, they knew that some of the conditions are hereditary, and some of the physicians were publishing syndrome. Syndrome means the combination of findings that have a recurring pattern. So like Down syndrome. And that was explained by Dr. Down, John Down, in basically 1862. And the same era that Darwin also presented natural selection. So, and the name Down syndrome basically coming from him. And the Dr. Victor McCusick was a brilliant geneticist and internal medicine internist, collected these syndromes that were basically published, and at the very beginning it was a small book, and then on my first year of fellowship it was a big book, and at the end of my fellowship it was 2 big books, then it was 3, 4, 5, and then eventually it had to go online. So at the beginning it was called Mendelian Inheritance in Man, in that catalog, and then online Mendelian Inheritance in Man. So when you see sometimes when you see a paper in the parentheses, McCusick number, OMIM number, or MIM number, that's this number in this catalog related to that disease. And OMIM, OMIM.org, it's the best source for having all the genetic conditions reported. You can have, you can search for something. You search for macrocephaly, it will give you all the genetic syndrome presenting with macrocephaly, intellectual disability. Another source of for you is genetests.org that explains the whole thing in regard to the presentation and the management and the genetics. Okay, and then about 80 years later, we had non-Mendelian inheritance. Like one example is Angelman syndrome, that we have kind of methylation, and it can be due to microdeletion, uniparental disomy. By now we know that the genes or the chromosomes have to come one from the father, one from the mother. If you have both coming from one of the parents, you may get some kind of autosomal recessive disorders, like we have it sometimes in cystic fibrosis, or you basically, if the gene on that chromosome or that region of the chromosome is imprinted, then you get that disorder. By imprinting, we mean that, again, when we look at the Angelman syndrome and Prader-Willi, both of them are caused by a deletion on chromosome 15q11.2. But if the deletion is on paternal chromosome, we get Prader-Willi. If the deletion is on maternal chromosome, we get Angelman syndrome. Why? Because the Prader-Willi on the mother's chromosome, Prader-Willi gene is imprinted, inactivated. The Angelman is imprinted as just stay active. So the same thing on father's blood. When the Angelman gene is inactivated, methylated, and the Prader-Willi gene is active. So if you have an parental disomy for maternal chromosome, in reality you don't have the Prader-Willi gene, you get the Prader-Willi syndrome. The other way around is if you have the deletion, because on the mother's chromosome, for example, for the Angelman region, because the Prader-Willi gene is not active over there, so you don't get Prader-Willi, but you get the Angelman syndrome, and basically that's the same thing happens when you have the deletion on the father's chromosome, you get Prader-Willi. So when we go to this, about 78% of those are because of those situations, a non-Mendelian situation, 1% about the chromosome abnormalities, 11% we have the gene mutation which follows the Mendelian inheritance, but putting together is about 90%, 10% of the situation still we don't know. But because we know how it happens, by a year ago when I was doing a presentation, a medical student asked, okay, if the gene for, say, Angelman syndrome stays active on the father's blood, what happens? And I didn't know. And I did search, and that's the basis of treatment of Angelman syndrome. Now we are doing that on the animals, but we activating the Angelman gene on the father's blood if it is deleted on the mother's blood, and in this way we can treat the Angelman. And it's going to be something in future. All right. So when we look at the chromosomes basically were known in the 1800th century, but it took at least 30, 40 years to know the correct number of chromosomes. We knew that it was called chromatin and mitosis. They knew about that, and the chromosome was the name was chosen based on the colored bodies, Greek word. And then Sutton and Bavaria, who were the basic, Sutton was a cytogeneticist and basically put the cytology with genetics together, and the study of chromosomes is called cytogenetics. And in 1912, Winnie Walther thought that the men have 47 chromosomes and women have 48. And then Painter in 1923 said that the number of chromosomes in human are 48. And it stayed in the textbooks for 30 years. And we should understand that they didn't have the culture, technology, or the staining or anything. They had to do it on the fresh cells, on dividing cells, like testicle cells. So he was getting the testicle tissues from the incarcerated prisoners and just smash them, putting on the microscope and counting the chromosomes, as if you put 23 cucumber or eggplant on top of each other and counting. And obviously you don't get a correct count. And then the name karyotype is basically was invented when you put the chromosome in order based on the length. And in 1952, Shu could draw the chromosome. When you're drawing the chromosome, it is called ideogram, all 48 chromosomes. And in 1953, somebody by mistake used a hypotonic instead of isotonic for fixing the chromosomes, and basically the chromosomes swelled and then separated from each other, and they could get a correct count. Not yet, but later. And colchicine can stop the metaphase, the cell division at metaphase. And basically, in 1955, Theo, who was American-Indonesian, was working at Shu lab. Also, Lewan, who was a Spaniard, and had worked in the Shu lab for a while and went to Sweden, asked Theo to join him and work on this Cauchy hypotonic technique, and he joined him in November '55, and in 3 months they came up with the correct number of chromosomes, 46. But having that in the literature for 30 years, they were very conservative, and basically they said, we do not wish to generalize the present finding into a statement that the chromosomes number is 46, but it is our finding that it is 46. And something that over for 30 years was a dogma, overnight was changed based on this kind of classic paper. And in the same decade, 1960, another agglutinin was discovered that causing the cell division. And these three, colchicine, hypotonic, and marginal, are used for cytogenetic study. So, and after having the ability to count the chromosome in 1958, Lejeune just found out that Down syndrome, 100 years after it was reported as a syndrome by John Down, that it is because of trisomy 21. And in 1962, he got the John F. Kennedy Award, and the Kennedys had an affected sister. And basically, after that, they could also report on 45X, XSX, XSY, XYY, and D group and E group, because at the time, still there was no banding. The chromosome identification was based on the length, and 13, 14, 15 were all same length, and 16, 17, 18 were same length. So they were just calling a trisomy one of the D group chromosomes. And this is different from Down syndrome. They had a child with anomalies, and they did the chromosomes, and then they found out that trisomy 13 or 18. And Noel and Hungerford in 1960, they discovered that the marker chromosome, the small chromosome in CML, is called Philadelphia chromosome because they were from that city. Basically, is associated with the CML. And Lyon, in the same kind of time, 1961, said that one of the chromosomes, X chromosome, is inactivated, and the bar body that was reported bar body by 1949 is actually inactivated X. And Lejeune, 3-4 years later, explained the macrodeletion chromosomes, pre-shot. But still, we were in the era of no chromosome banding, and in 9 years, only about 10, 11 syndromes could be identified until 1968 that Kasperson basically introduced the Q banding, which is a fluorescent dye, and you can see when you stain the chromosome, the bands, shiny band and dull bands, and basically you can identify the chromosome based on those bands. And then about 4 years later, the G banding, the IMSA banding was introduced, and we still use that. And fluorescent 10 years later, or 8 years later, fish, and 10 years later, comparative hybridization. So you see how fast everything now going. So it was the chromosome like this at the time of discovering trisomy 21, for example, like this, no banding, and based on the length, you could have a karyotype, longest number one is 22. In reality, the 21 is smaller than 22, but because it was already described as trisomy 21 with Down syndrome, we left it as where it is. And then with the Q banding, we can see the bands. The G banding, basically, without having the fluorescent microscope, we can have the study done, and then we could get longer and longer chromosomes. And on this chromosome, then eventually the cytogeneticists around the world just had a committee and meeting that how to call these different areas of the chromosome, the nomenclature, and there's an international system of cytogenetic nomenclature every 4 or 5 years, and they just introduced how to call it. For example, one example, the DiGiorgio Bellicordi facial 22q1 1.23 deletion, and each of these figures are presenting something. The first figure is a region, the second one band, subband, and sub-subband. So you don't read it as 22q 1123. You read it 11.23. So that double helix in 1953 by Crick and Francis, Crick and Watson, and basically we know that there are this is Crick and this is Watson, who was an American getting PhD in Cambridge, in Oxford, and we know that the two helixes are attached to each other with the base, adenine, thymine, cytosine, guanine, and adenine and thymine attached always together, and cytosine guanine together, and then we have the backbone deoxyribose DNA and the phosphate. Now, in the chromosome banding, actually the dark band on the G banding is AT rich bands, and they have less genes or no genes, and the light bands are CG rich, and they have most of the genes. So that's the reason we get different pattern of banding. So then about 10 years later, we had the fish, or not fish, just molecules using the radioactive, and then later on we could use the fluorescent to use to identify a specific region of the chromosomes. And the way it is done is basically you denature the chromosome, open up the helixes. You have a probe for that specific region, let's say the Prader-Willi 15q11.2, then you attach that probe to that strand, and basically if that area is TTT-ACC, you have the probe as AAA, complementary to those bases. And if you flag it with a dye, with a fluorescent dye, you can see them under the microscope. If they are not deleted, you see on the two chromosomes, both of them there. And basically, if you have a translocation, you have to probe in the two different chromosome, two sides of the break, you can even identify the translocation. And it is like chromosome 9 and 22 normal one, and then the translocated one, which is the Philadelphia chromosome. You can do it on the metaphase, which means that you culture the cells, you get the metaphases, and as if you are doing chromosome study, and do the fish on the slide, or you can do it in interphase without any culture. You can have it in 8-10 hours result. So if you suspect somebody has a Down syndrome, trisomy 18, you just send fish interphase for 21 or 18, and then next day you have the results. And otherwise, it takes about 3-4 days for the neonates. And if you have the fish probe for all the chromosome different colors, you get the spectra of the fish. And if you have all different probes as a cocktail for, let's say, MDS, multiplex syndrome, all different translocation on a chip, and you can do it just all of this all together. So comparative genetic hybridization, the name says, if you are comparing the genomic from patient and control, normal person, by hybridizing that with a probe, and if you color that probe, for example, in a patient in green and in the control in red, and you have exact amount of DNA, 1 or 2 micro, and exact amount, equal amount of the probe and the fluorescent, flag with the fluorescent. If they are equal number, there is no deletion duplication. Green and red together give you the yellow color, so there is no deletion or duplication. If you get more yellow, more green, you have duplication. If you get less green, you have deletion. And this is so basically simple. And you can do it all over the chromosomes, and that's the basis of doing the chromosome microarray. So you use the microarray, these copies of DNA, and then you flag them, and you just do the whole chromosome, the whole genome. Actually, you have probes and nucleotide that you can just all over the genome identify any abnormalities. Now, nowadays, when you have a patient, a baby with multiple congenital anomalous dysmorphic features, you don't do chromosomes anymore first. You do CMA as a first year testing, because most probably, if it is a chromosome abnormality, it is kind of unbalanced, and it's going to be picked up by chromosome microarray. And basically, as you see, all these different areas of the chromosomes can be just tagged, and then you can expand it up to the level of the bases, and you see which ones are missing. When you see the array study, there's a parenthesis, it gives you a number, 6-figure numbers, and that's the location of the gene or base pair that is missing or duplicated. And there are databases that you put that coordinate in that database, it will give you how much of the genetic material is missing or extra, which genes are involved, which genes of those genes are homing genes listed in the catalog, and if they cause the disease. So when you see the report, read it, because putting that report together after having the CMA, it takes hours and hours for the cytogeneticist to report that. And it gives you a lot of information. Or you just can send the patient to me. And then basically, it says you may use the oligonucleotide or a SNP, single base, and you can identify any imbalanced abnormalities, deletion, duplication, but you cannot identify the balanced rearrangement or translocation, as I explained in the technique. So uniparental guys, if you use this SNP array, you can identify, but not with oligo. And mosaicism less than 20% is not picked up by chromosome microarray. So that's important how to interpret this. Or sometimes, most of the time, you see the reports that it says variant of unknown significance, and basically this is the new testing, and we don't know all about the findings. They are going to be at the level of kilobases, or up to 5 kilobases can be picked up by this test, as opposed to karyotyping, the chromosome analysis, which goes to megabases that you can identify. So the level mosaic analysis said is important. Then, kind of in the same era, 1908, inborn error of metabolism was introduced by Sir Archibald Garrett, that also coined the expression of inborn error of metabolism based on the 4 metabolic disorders: alkaptonuria, benign pentazuria, albinism, and cystinuria, currently caused by defect in the metabolites, metabolism of intermediary metabolites and amino acids. And each was a lifelong condition and relatively benign. Each was transmitted as a recessive trait. This is the Garrett, and basically in most of the metabolic disorder, the substrate has to pass on from the membrane to the receptor or the transporter. If there's a defect in there, you get a problem, or the conversion of the substrate to different material, and basically if the enzyme is defective, you get all accumulation of the one before that and the problem related to that. So then in 1934, the phenylketonuria was introduced by Folling, and that's different from the other disorders that the Garrett has had explained, because this one gives you mental retardation, and basically it is also a lifeline. But if you identify the disorder by diet alone, you may prevent the mental deficiencies. So this is Folling, and the PKU basically started with him, and then in 1957, about 20 years later, newborn screening was introduced by Godfrey. But at the beginning, basically, he was also American, and he was a microbiologist, but in high school, he was the last student in 400 students in high school. But later on, just basically got different degrees in physics, biochemistry, microbiology in medicine. And in 1957, this Dr. Warner was working on developing a test for phenylketonuria, and basically helped him to develop that test in just 3 days. Basically, you put the blood from the patient on a paper and put it in the, he was a microbiologist, so in a dish that has bacteria is growing in it and has something to inhibit the growth. And then if you feed that bacteria with the phenylalanine that is food for the bacteria, that bacteria will start to grow and basically get positive results, and that was the Guthrie test. So at that time he called it just a bacterial inhibition assay, but later on it is a Guthrie assay. So Robert Guthrie, father of the mass newborn screening, why 4 years later his niece got the phenylketonuria, and he wanted to have a test that can be done on all newborns who pick it up, because if you pick it up, you can prevent the mental retardation. And he did. And now, basically, you can you do the the holistic, and the paper can be cut and put the same test on the dish on the bacterial growth, and you get the positive results. And basically, after that, in 1960s, it was kind of mandatory testing for a bunch of metabolic disorders. And later on, now that we do mass spectrometry, but Robert Guthrie, who also discovered other disorders, forever be remembered as a man who brought mass infant screening to every hospital in the world, not just US. And phenylketonuria can give you mental retardation, and kind of not very rare, 1 in 10,000. And the thing about the phenylketonuria, the treatment is lifetime these days, because if the mom, they used to just stop the treatment with diet when you get older, because it didn't have any adverse effect. But if the woman is affected getting pregnant, that will affect the baby, even if the baby doesn't have the disease, high phenylalanine. And the metabolic disorders in general involve around metabolism, so usually you get them with acute or chronic encephalopathies. So one of the reasons of acute encephalopathies other than these reasons can be metabolic disorders, and this present with hyperammonia and most of the time acidosis, and you have a baby, you suspect metabolic disorders, you order 3 tests screening for metabolic disorders, plasma amino acid, urine organic acids, plasma carnitine-free and total, and acylcarnitine. And you just at least look at 90% of metabolic disorders. And in chronic encephalopathies, you usually see the babies with developmental delay or seizures. If there are no abnormality outside the central nervous system, then it is probably a syndrome. Then if there are no associated findings, then you have basically signs of gray matter disease, seizures, and visual impairment. And again, by doing those tests, many of them can be picked up. So back to the Philadelphia chromosome and Noel and Hungerford. Basically, it started the process that the cancer is not caused by virus or something. It is a genetic condition, and there's many of them. We have translocation of the chromosomes. And that was 1960, about 12 years later. Generally, at the time they were seeing the chromosome, the marker chromosome 22, didn't know it is a translocation. Generally then discovered that it is translocation between chromosome 9 and 22. And basically, she discovered many other translocation-related cancer, and those from Bill Clinton and Obama. And then 30 years later, we could, based on that findings, okay, this translocation, there is a thymidine kinase that stimulates the production of the fusion protein. If you have something to inhibit that thymidine kinase, you have the treatment. And in fact, Gleevec for CML is that treatment. And 95% of patients respond to that treatment. So nowadays, the treatment is based on going after the mutation and what that mutation does, and then have the treatment just blocking that the effects on that mutation. So it is at the molecular level. So and now we have also the same technique, the treatment for melanoma and a handful of other cancers. So, and then we found out that these translocations and extra chromosome or missing chromosome basically are associated with the cancer, each of them are meaning something. The translocation always not causing the fusion gene that causing problem. For example, chromosome 8 has an oncogene MYC on it. When 8 is translocated at that point, the immunoglobulin genes on 2 light chain or heavy chain or copper chain on 2 or 14 or 22, you get the promoter of that immunoglobulin gene stimulates the MYC gene, which is an oncogene, and you get the cancer. So basically, the genes can be either oncogene or tumor suppressor gene. If you lose the tumor suppressor gene, you get the cancer. If you get amplified oncogene, you get the cancer. It is like in your car, the gas will be oncogene. The more you push on the gas, you get higher speed of the cell division here. And the same thing, if you just don't use your braking the slope, then basically you get it in speed and cell division. So it's not just always one gene, oncogene or subgene. There are multiple genes involved. It's like an environmental factors, like the same car in the slope, and the environmental factor that the slope causing more speed, and you push on the gas, a car from behind you push you down, and somebody pulling the car with a cable from the front. These are all different genes that basically work together to cause leading to cancer, or some types of cancer. And many of the hematological malignancies are acquired, so you are not born with that gene defects. You may get it later on the somatic based on translocation or mutation. And just so you know that the stem cells give you the myeloblast stem cells and lymphoblasts goes to lymphocytes. But the myeloblast makes all 3 cells, the platelets, the red blood cells, and the white blood cells, nucleotides, and basically different type of cancer. So the DNA alteration can be at the DNA or RNA level, and DNA methylation, changing chromosome number, loss of heterozygous chromosome translocation, gene amplification. Okay, going from the deadly disease to life, and basically this is in prenatal genetics. You either have screening. By screening, you just change the risk of a person or the patient or woman in regard to having a specific condition. By diagnostic tests, you actually do some invasive testing in pregnancy, like amnio or CVS or fetal blood sampling, and to make a diagnosis. And you know, the screening that can be ultrasound, your nucleolucency that is associated with Down syndrome, or first trimester markers like maternal serum markers, and the PAP and HCG, and also the second trimester you have triple testing for quadruple testing and PAP. And then when we look at the history in just 1970, 50 years ago, we started to have amniocentesis based on age alone. And then in 80s, 10 years later, we had these screening testing that changes the risk, and based on that, you do diagnostic testing. And then also first started with second trimester testing and then first trimester testing added. And later on, just 5 years ago, we have non-invasive prenatal diagnosis, but through the fetal DNA, I will go over it. So at the time, still advanced maternal age, previous child with defects, stillbirth, multiple miscarriages, and the screening abnormal, you do the diagnostic testing. So when you do the amnio, about half of the chromosome abnormalities that you have is still Down syndrome. 13% is 18, and 5% 13, and about 13% is sex chromosome abnormalities. So by doing just this 5 chromosome screening, you are looking at 85% of abnormalities. And that's why sometimes when you have the ultrasound and the baby shows some abnormality, you just send a fish for these 5 chromosomes and you get 85% of abnormalities picked up. And at the beginning again, at the 70s, 5% of women were over age 35, accounting for about 30% of all Down syndrome born. And then because of women getting into career and the workforce, and 20%, instead of 5% getting, were over 35 to get pregnant. 50% Down syndrome are related to the age now. And these are based on those markers, which one is up and down. You can say which of these is associated. NIPS, non-invasive prenatal screening, it is through the fetal DNA circulating in the mother's blood during the pregnancy. It is new, just 5-6 years ago, it started to be used. But at the beginning of 1997, they discovered that the cancer patients have the DNA from the cancer tissue in the serum, that person's serum, and basically they applied that to 30 pregnancies in the male pregnancies, and they had the markers for the Y chromosome. They could identify their sex through that technique, through the fetal DNA circulating in mother's blood, without any amnio or anything. Correctly, the sex was identified. And then eventually, the next generation sequencing, which is massively parallel sequencing, is called. They have the sequences from different chromosomes. So you have, if you identify the sequence in the mother's blood and the fetal DNA, you know which chromosome it belongs to. Right now, for chromosome 13, 18, 21, X and Y, they do this testing on maternal blood through the fetal DNA, and they can pick up the DNA, the trisomy 21. So if you have 3 copies of that sequence of chromosome 21, you have trisomy 21. So basically, these are different type of screening that we do now. And if we go with just absorption alone, the detectability is 64, 70%, up to 98% that now is cell-free fetal DNA testing. So the screening alone, again, will pick up about 88, 99, but that's a combined. When you use both ultrasound first trimester, second trimester screening all together. And this has a higher sensitivity. And you are going to get these more and more. So now, ACOG is now saying that age alone is not a factor for doing the diagnostic testing. Even the screening test should be offered to everybody. And also, it's better to use all the means available for screening, ultrasound, first trimester, second trimester screening. So then we have now the technology of pre-implantation genetic diagnosis. And which means that after you have the conception and the zygote and the cell division, the blastoma to up to 4 or 8 cells, if you take one of the cells out and do testing fish or molecular study for a disease, let's say cystic fibrosis, the parents are carrier, you can do this before the actual after the pregnancy, you check the embryo. If it is normal, not carrying both genes abnormal, you can put the embryo in. And basically, you stimulate the egg formation here, and then you do X's, which means that intracytoplasmic injection of the sperm, and then you get the blastomere. You can take one cell out and test it, and then in the last 3 days you can put 3 embryos after testing. So these are different methods, and so going to the past and present, sequencing, and our future still digging and finding new findings, and our future Newtons are using Apple iPhone and to discover new things, and still we may see odd things like President Trump. So 2099 or sometime in the future, are we going to be computers? Computing, most possibly. So everything is going to be computerized, and you go to testing, and right now most of them are. And thank you for your attention. Now, so these guys are not. And I don't know, do we have time for questions? I don't think so.
It's after 1.
It's after 1, so there's no time. Okay, they can read it.